Hello everybody and welcome back or welcome to my channel. This is Ruhan or the Tilted Hypotenuse as you see on YouTube. You can follow me on Instagram. I have the link right here below in the description. Let's start. The fungi kingdom. So what exactly is the fungi kingdom? They're multicellular organisms. They're multicellular. That means they have more than one cell in the whole organism. They're eukaryotic, which means that they have cell organelles. Unlike prokaryotic, unlike bacteria, unlike Monera, they do have cell organelles. And they're heterotrophic. Heterotrophic is a wide term which is given to many organisms. It means that they eat other organisms for nutrition. Autotrophs, like your algae, like your plants, like your some types of animals also, they're autotrophs like a snail. There's a special snail which has chlorophyll A. So these type of organisms, they make their own glucose. We cannot make our own glucose. We need to eat something. We need to respire to create our glucose. So the most common examples of fungi kingdom are Saccharomyces and Penicillium. So Saccharomyces is the scientific term for yeast and Penicillium is penicillin. This is how yeast looks like under a microscope. And this is penicillin's chemical structure. It's a very complex chemical. It's a medicine. This R stands for the alkyl group. So it's continued this way. And sulfur, nitrogen, oxygen, methyl groups, hydroxyl groups, oxygen. This shows that it's a very complex chemical and it's created by penicillin. Next. So here we have something called a mycelium. What exactly is a mycelium? It's a structure on a fungus that is a network of thin projections. And those thin projections are called hyphae. What a hyphae does, it's like a modified form for the fungi to get its nutrition. There's two types of hyphae. There's a septate hyphae, where basically how a septate hyphae looks is like this. These are the two types of hyphae. One hyphae can never have septate and aseptate. That's like a very key term to know. One type of fungus will always have septate hyphae, and the other one will always have aseptate. On the same mycelium, you cannot have a septate and an aseptate hyphae. So septate looks a little like this. There's breaking points here. And there's a nucleus inside each of them. And each of these nucleuses are diploid. Aseptate is more of a cenocytic type of hyphae. The septate is in proper sections. Aseptate hyphae, it's a cenocytic type of hyphae. So a cenocyte is basically an organism that has karyokinesis taking place, but not cytokinesis. So we all have seen what binary fission is. One cell divides into two. This is a very common type of example. So this breaks into two. So here what is happening is that this nucleus which is diploid is giving rise to two diploid nuclei. Okay. And cytoplasm is splitting into two. So here you get the first cytoplasm, here you get the second cytoplasm part. So when a cytoplasm splits into two, it's called cytokinesis. And when a nucleus splits into two, it's called karyokinesis. So here what's happening is karyokinesis is taking place. So you get all your nuclei here. But you don't get your cytokinesis taking place. So this hyphae, it's not splitting into many parts like you see in binary fission. So this is called cenocytic. There's no end breakpoints. Here you can see septates here. There's no septates present here. It's just nuclei. So these are the two types of hyphae. This is septate hyphae here. It's growing on a strawberry. So next, this is called mycelium. Now you have fungal modes of nutrition. Just like we need to eat other organisms for nutrition, fungi have a different form of nutrition. So what exactly happens here is, whenever I show a pink slide here, it shows that you have more than one type of diagram present. Give me 1 second. Yes. Whenever there's a pink slide here, a pink slide is present, there's going to be more than one type of nutrition present. So here, let's start. The first type of nutrition you see in a fungi is saprophytic nutrition. Saprophytic nutrition is always present in fungi. So what does saprophytic nutrition mean? It means that the fungus you have is feeding on dead matter. When a fungus feeds on dead matter, it's taking nutrition from the dead organism. It's organic matter. After that, you have parasitic forms. Parasitic fungi are the types of fungi that live inside other organisms or on other organisms, and they feed on their organic matter. So it's a different type of saprophytic form where it's feeding on living organic matter, or it's stealing nutrition from the other organism. And then you have kind of a positive mode of nutrition called symbiotic nutrition. Yes, venom and Eddie Brock, they're symbiotes. So fungi also have symbiotes present. We'll learn about symbiotic forms. There's two types of symbiotes here. There's lichens and there's mycorrhiza. Symbiotes are basically types of nutrition where both the organisms involved get some benefit out of it. Okay, there's some sort of benefit you get. If I have a symbiote with me, that symbiote's going to get something from me and I'm going to get something from them. So your first one is a lichen. What exactly is a lichen? It's a symbiotic relationship between lichen. A lichen is a symbiotic relationship between fungus and algae. So here is a lichen. It's a symbiotic relationship between a fungus and an algae. The large surface area of the hyphae on the fungus, it can be either septate or aseptate. It helps gather nutrition for the algae. So basically how it works is, you have your algae here. This is your algal component. And then you have your fungal component here. So this is how a lichen looks like. This is your fungal component. This is your algal component. So we all know that algae are green. There's three types of algae. So there's chlorophycae, rhodophycae, and phacophhycae. Oh, right now, the algae we're talking about is chlorophycae, which is green algae. They have chlorophyll A and B. Chlorophyll A helps in photosynthesis. So the first part here to note is that this algae is conducting photosynthesis. It's creating its own food. All fungi are heterotrophic. That means they cannot create their own food. So here what happens is, the algae photosynthesizes and provides the heterotrophic fungus with its nutrition. So that heterotrophic fungus is getting its nutrition from this algae. And the large surface area of the hyphae, this hyphae here present, it's helping the fungus gather nutrition for the algae. What do you need for photosynthesis? You need carbon dioxide, you need water. This fungus can gather that. It has that type of ability. So this is called a lichen. This is a photo of a lichen. So the red part you see here is the algal component, and the gray matter you see here is the fungal component. Mycorrhiza is let me clear these annotations. Mycorrhiza is another type of symbiotic relationship, and it's between a fungus and the root of a plant. So this is easier to remember. because it's between something that you see every day. You see mycorrhiza. The roots of a plant and the fungus together work symbiotically. How do they work though? So this root of the plant, what it does is it collects water. It needs water, xylem, phloem, transport. For all of that, water is needed for many functions carried out. It's to cool the plant down. It's for photosynthesis. You need water for turgidity. So a lot of stuff water is needed for. So you have your fungus here. Can you see this fungal component? And this is your root component. So there's a documentary on Netflix which talks about mycorrhiza and about lichens. It's called Fungi. So you can watch that before watching this video. This video is more on a academic concept. That video is more of a casual view. So here the fungus supplies the plant's root with water and minerals. Root can take water and minerals by its own, but fungi has chosen to come here. The plant roots sink to the fungi with glucose from photosynthesis. So translocation is present here. The sucrose formed from photosynthesis, it gets converted to glucose. That glucose will come here to the fungi, and that fungi will use it for nutrition because it's heterotrophic. Next, lichens and mycorrhiza is clear. Next, we have the types of fungi. So there's a total of 4 types of fungi. They are Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. They're really easy to know. Deuteromycetes are more of imperfect fungi. They're not a specific class. So 3 of the photos are here. Phycomycetes look like these. Let me take my video away. Phycomycetes look like these. Ascomycetes look like sacs. And Basidiomycetes are your simple mushrooms. Phycomycetes. They're the most primitive fungus on this planet. The oldest fungus you know is Phycomycetes. Their hyphae is aseptate and cenocytic. So out of all the fungi present here, Phycomycetes are the only ones which have aseptate and cenocytic hyphae. All of the other ones have septate and branched hyphae. So let me show you a drawing of how this fungi looks like. So its hyphae looks like this. It's aseptate and it's cenocytic. They grow in moist and dense places. All fungi grow in moist and dense places. Wherever you see a fungi present, that place is going to be moist, that place is going to be dense. On your house, in your interior, when you see there's green fungi growing on the walls everywhere, it ekes you out. That's because those walls aren't clean. So they're moist, they're dense. So udhar, fungi will grow. And there are 2 types of, there's actually a total of 3 types of sexual reproduction that's present. It's isogamous, anisogamous, and oogamous. So isogamous is the form where both your gametes, where both your sexual nuclei, the body which holds the nuclei, isogamous mein, they both are of the same shape, the same size, the same type, except for the genetic material inside. So you cannot differentiate when there's isogamous nuclei present. So isogamous, this is the male isogamous gamete, this is the female one. You cannot differentiate between them. Anisogamous, you can differentiate between them. So here is one anisogamous male gamete and here is another anisogamous female gamete. So you are able to differentiate. The shaded one is the female part. Oogamous is a very, very extreme form of anisogamous. So for oogamous, what happens is you have your sperm cell, very differentiable, and then you have your egg cell, which is very large. It has special organelles, corona radiata, zona pellucida. Sperm cell has flagella, it has acrosome. So there's many parts. So you can differentiate it easily. So this is what Phycomycetes is. So let's go ahead. You have your examples for Phycomycetes. So what I do is I create these small acronyms, RAM, random access memory, the god RAM. So the easy way to remember this is Rhizopus, Albugo, and Mucor. These are the 3 Phycomycetes you need to know. RAM. So asexual reproduction in Phycomycetes. How does it work? How does asexual reproduction in Phycomycetes work? So what happens in asexual reproduction for fungi is that, especially in Phycomycetes, what happens is that you have the hyphae. Hyphae are involved. Hyphae is not only for nutrition, there's other many aspects for which hyphae is important. So here what happens is you have 2 hyphae present. They're from different Phycomycete fungi. Both of them are aseptate. So you have both your hyphae. What happens is they want to get lucky today. They want to mate. So here this is asexual. There's no fusion of gametes involved. There is karyogamy involved. Fusion of nuclei is involved, not fusion of gametes. Sperm and egg fusion is sexual. Conidiospore fusion is sexual. There's many other stuff. But here, only nuclei are fusing. Gametes are not fusing. So what basically happens here is that this hyphae A, there's no male and female here. As I said, there's no sexual reproduction. They grow these projections, they're finger-like projections. So they grow and then they join together. They form a special body called a zygosporangium. This special body is called a zygosporangium. Okay. And what happens here is that in this zygosporangium, plasmogamy takes place. What exactly is plasmogamy? Plasmogamy. Plasmogamy is the fusion of the cytoplasm. It's that easy. It's the fusion of the cytoplasm. So here, this is the cytoplasm, the white portion you see on the hyphae, this complete white part, that's your cytoplasm. So here they're fusing together. यहाँ plasmogamy हो रहा है. So both of the cytoplasms fuse. They create a new body called a zygosporangium. And in that zygosporangium, what happens next is this is your zygosporangium. You have your first hyphae here. You have your second hyphae here. So here what happens is, so there's plasmogamy and there's karyogamy. So here you have your first, I'm drawing 3 nuclei of each. So here you have your first ones and here you have your second ones. These nuclei, they're haploid in nature. N. Haploid is denoted as N and diploid is denoted as 2N. D, diploid 2, haploid 1. Triploid is also there, but for right now we don't have triploid. Triploid is in plant kingdom. There's dikaryon also. So here what happens is these nuclei present, they fuse together. So here you have your nuclei, they fuse together. So you get 3 cells, they're fused. I'm not shading them, I'm creating this. This is a new type of body. Now it's simple algebra here, n plus n. 1 plus 1 equals 2n. So here you get diploid cells, diploid nuclei. The zygosporangium is still present. So what happens next is that once these you get these diploid nuclei, these diploid nuclei divide by meiosis. So what happens here is that your zygosporangium is present, you get meiosis taking place. So I'm clearing this whole drawing. So here you get your zygosporangium. Another body called a sporangium, a simple sporangium, forms on the zygosporangium. So this is called sporangium. So on this sporangium, what happens is you have a special type of these diploid cells. They travel up here. So they go here and they form this body called a sporangium. So after this, what happens is endogenously, haploid spores are created. Meiosis क्या होता है? 2n gives rise to 4n, 4, n. So 2n will split into 4. So one diploid nuclei will split into 4 haploid nuclei. So you get your haploid nuclei here. There were 2 haploid nuclei. So you get 8. So you get 8 haploid nuclei. So there were 2 2n nuclei. So all of this gets multiplied by 2. So you get 8 haploid nuclei. These haploid nuclei are called zygospores. What exactly is a spore? It's just a body that has cytoplasm and nucleus. We label this spore. And here you have your nucleus. So this is what a spore looks like. So you have your sporangium, you have all your spores present. This nucleus is haploid, okay? जो Phycomycetes था, the Phycomycete which I started from the hyphae, their nucleus was also haploid. So this spore is also haploid. So when the spore will germinate, when it will grow back, it will grow into a haploid fungus. So now this sporangium is filling with many haploid nuclei. Just because the zygosporangium created, because of 3 nuclei haploid, you got 3 cells. These 3 divided into haploid nuclei. So 3 raised to something. So here you got your 8 haploid nuclei. All of these 8 haploid nuclei create something called a spore. These spores are called your zygospores. So what happens? This sporangium, it fills up with these nuclei and it will finally burst. It can't withstand that amount of, that amount of population of spores growing in there. So once this bursts, what happens is that you get your spores and then they germinate in appropriate conditions, moist and dense conditions. So this surface, this is very moist, this is very dense. So your spore will plant here. This is your haploid spore and it will grow into another hyphae. And this is aseptate and cenocytic. Okay, mitosis say it will divide. So a simple rule of mitosis and meiosis is that 2n, when you're conducting mitosis, you get another 2n cell. Okay, 2n will split into another 2n. 2n will give 2n. So this is how mitosis cycles. So the ploidy of the nucleus doesn't change. In meiosis, the ploidy does change. It turns from diploid to haploid. Haploid cells will always conduct mitosis. Okay, it's a rule of thumb. Haploid nuclei always conduct mitosis. And diploid nuclei always conduct either mitosis or meiosis. So this is a rule of thumb. Haploid ones will always conduct mitosis and diploid ones will always conduct meiosis. So this is how the whole asexual reproduction in Phycomycetes work. So this is how your asexual reproduction in Phycomycetes works. So now you have sexual reproduction in Phycomycetes. Asexual, zygosporangium forms, you get your sporangium from there. There's a lot of meiosis and mitosis taking place. The sporangium bursts and the spores germinate. For sexual, It's a little more complex. So what happens in sexual is, so let me open a whiteboard. So here what you see is the whole sexual cycle. Now, for asexual, what happens is nothing. There's only two nuclei. So you have your hyphae here. They just join. The whole fungi which you have, it joins together. And that is your asexual reproduction. So the keywords in Phycomycetes reproduction is sporangium. What exactly is a sporangium? A sporangium is a body which holds haploid spores. It holds haploid spores. Diploid is a nucleus which has two N formation, and haploid is a nucleus which has single N formation. Plasmogamy is considered as, what is plasmogamy? It's the fusion of the cytoplasm. And karyogamy is the fusion of the nuclei. Karyokinesis and cytokinesis is the opposite of these. Plasmogamy's opposite is cytokinesis, and karyogamy's opposite is karyokinesis. So here they're joining together. In karyokinesis and cytokinesis, they split together. So this takes place in binary fission. So karyokinesis is one nucleus and it's splitting into two. While cytokinesis is a cytoplasm which splits into two. And in cenocytes, cytokinesis doesn't take place. So what is a zygosporangium? The zygosporangium was the body formed when the two hyphae created projections and plasmogamy took place. So un dono ka cytoplasm, it fused together and it created a zygosporangium. Endogenous growth and exogenous growth. This is very simple to know. Endo means inside, exo means outside. Exothermic reaction, reaction which gives out heat. Endothermic reaction, reaction which takes in heat. So same here, endogenous is a growth which happens inside the cell or inside the cell organization, and exogenous is outside. So here what did you observe? It was endogenous growth. Inside the sporangium, inside your sporangium, the zygospores were formed, the haploid spores, and then they burst out. So here you see endogenous growth. Exogenous is not observed here. Exogenous is basically the spores forming outside the cell. Zoospores and aplanospores. Again, it's very simple. Zoospores are spores which can move around. Okay, they're motile spores. And aplanospores are spores which cannot move around. So zoospores are motile spores. And aplanospores are non-motile spores. So an easy way to remember aplanospores is aplan. A means not present. Asexual. There is no sexuality present there. So aplanospores are spores without a plan. So they didn't make a plan to move around. So that's aplanospores. Ascomycetes. This is the second class of fungi. They're known as the sac fungi. So what exactly is sac fungi? They're septate and branched. So this is how they look like. They have septate hyphae. They have branched hyphae. So we know what septate is. What is branched? Nothing. Branched is just like a tree branch. So more hyphae will branch out of one hyphae. So this is how your ascomycete looks like. This is how an ascomycete looks like. And the most common fungi are present here. You have your penicillin here, which is multicellular, and you have your unicellular, which is Saccharomyces. So Saccharomyces is yeast. So you have your Saccharomyces, which is the only unicellular fungi. in the whole kingdom. Only one unicellular fungi is there, which is yeast. If it's a special fungi, then it has to be something special. They have a special type of nutrition. It's called coprophilus. Let me turn on the laser pointer. So what happens here is you have your saprophytic 1. It's a decomposer type of nutrition. It feeds on dead matter. There's a dead body lying around, there's a dead animal, a dead plant, a dead person. So saprophytic fungi will grow on the dead animal, on the dead matter, and it will feed on the dead matter. Dead plants can also work as dead matter. When a dried piece of wood is there, that wood was once living. It had living cells in it. Now it's dead. You have parasitic, feeds on living organic matter. Most disease-causing fungal infections which are there, they're parasitic. So you get eczema, you get fungal eczema, scalp pe fungus grow hota hai. You get skin eczema. There's fungi spores. So your skin, when you don't clean your hair, when you don't take proper baths, when you have an unclean scalp environment, your scalp turns moist, it turns dense, it turns oily. So the spores which are there, spores are very small, they're not visible, they are all around the air. So when you see a dry and oily scalp, a dense, a moist scalp, the spores will germinate on your head. The spores will germinate in your scalp. Then they will take nutrition from there. So skin turns dead, it shrivels up, it dies, then you get dandruff. So that is parasitic fungi. And coprophilous fungi, name is very big, but then learning it is very easy. It feeds on dung and excrement. So this is a type of fungi which grows on excrement, on excretions, especially dung, cow dung. So this is another type. Next, you have asexual reproduction in Ascomycetes. So what basically happens in asexual reproduction of Ascomycetes is you have one type of Ascomycete. Its name is Neurospora. Okay, we will learn Neurospora in examples. There's a lot of Ascomycetes. For us, we just need to learn 3 of them. This is NCERT. So NCERT has only 3. There's Neurospora, Oscillatoria, and Anavina. So here, what happens is, Neurospora, Oscillatoria, and NOA, Aspergillus. There's a very easy way of learning this asexual reproduction. What happens here is that you have your Neurospora. It's branched and it's septate. This is asexual. There's a lot of Ascomycetes. There's claviceps, there's Aspergillus. Neurospora is used in genetic research. So here you have your Neurospora. It's branched, it's septate, and it's haploid. So the special hyphae, this type of hyphae only asexually reproduce karne ke liye when there's hyphae created. It's called a conidiophore. This hyphae is called a conidiophore. Now exogenous growth takes place. Okay, the haploid only. This is haploid N. Exogenous growth will take place on the conidiophore through mitosis, because haploid ones only conduct mitosis. And you get your normal spores. That is it. And these are all haploid. They will fly around. The wind will carry them. And they will turn into Neurospora again. So this is called your conidiospore. So this is called a conidiospore. Once you get your conidiospore, it will travel around in the air, it will find a good place to germinate, and then it will turn into another Neurospora. That Neurospora will be haploid, it will be septate, it will be branched. After this easy type of, this is very easy. This is also exogenous. So this is exogenous spore development. Next, you have your sexual forms. Okay, sexually reproduction जब भी होता है, that time what happens is this whole process takes place. You get your conidiospore from mitosis, the whole conidiophore forms, you get your conidiospore. What later happens is, we have to zoom in onto one conidiophore now. So here, the conidiophore which you get, I'm going to clear the drawing. So you get one conidiophore here. On this conidiophore, you have a conidiospore. So let's imagine that another spore from another neurospora landed on this one. Now they both are conidiospores from different neurosporas. So what happens is, here again, plasmogamy takes place. Both these spores, they'll conduct plasmogamy. So let me zoom in on this. You get a body like this, and your this whole membrane which is there, it breaks down and you get plasmogamy here. So cytoplasm will fuse. यहाँ पे nuclei are not fusing. Karyogamy did not take place. Only plasmogamy took place. So this special body where only plasmogamy took place, this is known as your ascus. Ascus is your plural. This is known as an asci. So what happens here is, karyogamy will take place, but it will take place a long time in the future. It takes up to more than 100 years. Sometimes. It's a very time-consuming process. Here you have your asci formed. So with this asci, what happens is, we've learned diploid, we've learned haploid. So this is your diploid, this is your diploid, this is another diploid nuclei. This one is also diploid. No meiosis has taken place. Haploid ones can't conduct meiosis. So this will stay diploid. But they haven't fused yet. Ye hona baki hai. So what happens is, you get some form known as a dikaryon stage. So what happens in dikaryon stages is, it's called a dikaryon phase. So it stays in N plus N form. It doesn't turn diploid yet. This is your dikaryon phase. It hasn't turned diploid yet. So what happens here next is that in this ascus is known as a dikaryon. So ascus is dikaryon now. After this, what happens is, let's go 100 years ahead now. Fusion ho gaya, karyogamy took place. Now what happens is, you have 2N nucleus. So you have your neurospora here. Your ascus forms and your karyogamy takes place. So here you have a 2N nucleus now. N plus N finally took place. It didn't stay at N plus N. I'm learning how to use the Apple pen. Okay, so you have your ascocarp. So what is this thing called? Ascocarp. And there's more than one forming everywhere. Here one spore formed, here another one lands from another neurospora. There's one more conidiospore formed here, another one will land here. So a lot of ascospores will form, ascocarps will form. So you have one more here. You have one more here. Okay, so all of these are diploid after karyogamy took place. This is sexual reproduction. So here what happens is, again now this is diploid, 2N meiosis, 2N gets converted to N. This 4 ends formed. So here you have this. This 2n will give rise to 4. All of these are haploid. And each of these will conduct meiosis. Okay. Each of these will conduct mitosis. Mitosis mein 1 gives rise to 2. So here 1 is converting to 2. So here what happens is, this can't conduct meiosis. This will conduct mitosis now. So you get a longer ascocarp. So this one splits into 2. This one splits into 2. The third one splits into 2. The fourth one splits into 2. So you get this very big body. And this big body is called your ascocarp sac. Sac fungi. This is where the name sac fungi came from. So you get your whole 8 haploid nuclei. These all are N. Mitosis always gives rise to same ploidy. Now, nothing will happen. This ascocarp will burst. Spores are released. So this spore, they'll turn into spores now. They have a little development going inside. They develop a little from nuclei. They grow cytoplasm. So once the spores are formed, the ascocarp sac gets the pressure from inside, the pushing pressure, it's too much. So it bursts and then it releases the spores. These spores are what? They're haploid. Now they land anywhere. For example, this spore lands here. So this is called, these things are called your life cycle. This is your ground. This is any surface that is moist, dense, and habitable. This haploid, this thing is called a ascospore. This spore which is released, it's called an ascospore. So Ascomycetes, ascus, ascospore, ascocarp, sac. So everywhere you see the prefix asco. So this is how the cycle takes place. So there's a lot of stuff happening here. You get your spores, your ascospore. It's haploid in nature. Another spore lands on this one. They conduct plasmogamy. Karyogamy doesn't take place for around 100 years. Once it does take place, you get a large sac kind of structure. Haploid nuclei conduct meiosis. It gives rise to 4 other haploid organisms. These haploid nuclei will conduct mitosis and they will split into 2 each. So 2 4s are 8. You get 8 total spores here. There's a lot of pictures of ascocarps. You can see 8 large nuclei formed there. Once this ascocarp will burst, your spores will get released, your ascospores, and they will germinate, create a new neurospora, and the whole cycle will take place again. So this was sexual reproduction in Ascomycetes. So now that we did sexual reproduction, let's see all the Ascomycete reproduction keywords. You have your conidiophore. What is a conidiophore? It's a special branch that was created in asexual reproduction. It's a special branch that will conduct mitosis and make conidiospores. Conidiospores are they're the parent spores per se. They're the ones which fuse in sexual reproduction. They're haploid. Conidiophore is haploid. Exogenous growth outside the body. So these conidiospores are forming outside the conidiospore. Ascus, it's a dikaryon phase where 2 haploid nuclei haven't fused yet. They're ready to fuse. This can take place for over 100 years. So your ascus is present. This ascus is a special structure. It has plasmogamy conduced. No karyogamy is taken place. Then you have your ascocarp. What an ascocarp does is, ascocarp is the sac. It's the ascocarp sac. It conducts the haploid nuclei. It holds the ascospores. Ascospores are haploid. They germinate under the right conditions and then they create a new ascomycete. So there's this, to learn the examples, there's a very special kind of concept created by many teachers worldwide. It's called as the sac concept. So this is sac fungi. So it's called the sac concept. It's quite funny. You're going on a camp. You're going for a camp alone in the forest. You're going to take a sack with you on your bag. So obviously, we get hungry fast. We need bread. So instead of taking bread only, we can take yeast. We can create yeast there. Yeast will create bread for us. So yeast is the first example of Ascomycetes, Saccharomyces. You're thirsty, but the forest is too much to handle. You're sober right now. Yeast will give you alcohol also. Yeast is pretty kind. It created alcohol for you. Now you're kind of hungry. After eating all this bread, the bread soaked up the alcohol. So you're hungry now. You have morels and truffles with you. You took your sack, you were hungry, you've taken a, I don't know, nowadays there's a lot of diets taking place. So you've taken a Ascomycete diet where you can only have Ascomycetes, you can only have fungi. So here you get your morel and your truffle. Both of these are very large delicacies. This morel looks very unappetizing, but it's considered a delicacy. And your truffle, we've heard chocolate truffle, so it's not connected to this. The chocolate truffle is it's a piece of chocolate which looks like a fruiting body. It's called a truffle. So you have your morel and truffle, and then after eating all of this stuff, too much fungi you ate, too much beer you drank, you fell ill. Now you need penicillin. So luckily you found a pocket of penicillin with you. So you have your penicillin medicine with you. So these are the 5 examples of sac fungi. There's yeast, there's morels, there's truffles, and there's penicillin. After this, you have one more type. You have ACN, which we just did. So for ACN, what happens is you have Aspergillus, you have Claviceps, and you have Neurospora. So let me just write that down here. You also have Aspergillus. These 3 you have to learn by heart. This is some kind of thing which you can, remember, you can visualize that you've taken yeast with you. Yeast packets come everywhere. So you have all of this present. Aspergillus, Neurospora, and Claviceps. Neurospora you can remember because we did asexual and sexual reproduction of that Neurospora. Aspergillus and Claviceps, these are still important. So you still need to know their names. After this, what we have is Basidiomycetes. What are your Basidiomycetes? They're the most common thing you can visualize after listening to the word fungus. So Basidiomycetes are the mushrooms, bracket fungi, and puffballs. So what happens here is that they grow the best in soil, on logs, on tree stumps, and they sometimes grow as parasites. I'm not aware of mushrooms growing on people. So parasite, you have to remember it that way. This can be a plant parasite also. So soil, logs, tree stumps, parasites. So their mycelium is also branched and septate like Ascomycetes. One lucky thing for you here is that there are no asexual spores formed. Asexual reproduction doesn't take place. So there's one less thing to learn. These are the 3 types of fungi you see. So here what happens is you have your mushroom. Mushroom looks like this. Okay, this is a simple mushroom. This is your agaricus mushroom, common garden mushroom. Then you have bracket fungi. So have you ever seen a tree and then there's this type of umbrella like fungi growing on it? They look like stairs. So this is known as your bracket fungi. And puffballs, if you play Mario Kart, you might know what a puffball is. You can Google the image. It's a red colored with it has a lot of a lot of boils on it. It's red colored and white boils on it. So that's known as a puffball. These fungi you can see over in Mario Kart game. So this is puffball. After this, you get fragmentation. Because there's no sexual reproduction, God decided to create something new for them. So this is a special type of vegetative reproduction. It's called vegetative fragmentation. So here there is no spores, nothing, there's no fusion, there's no nuclei, haploid, diploid, nothing. This is your normal basidiomite. It breaks into two. You kick it. You're being evil. You break it into two. So it splits into two. So this one is still here and this one falls down. This is the ground. This one falls down. This is called fragmentation. After this, what happens is on your ground, this one will grow into another fungi. And the one which fell down, he doesn't want to die easy. So he'll also grow another fungi. So this is how fragmentation takes place. It's this easy. You break it into two, another piece falls down, the piece grows into something else. So this is. So after fragmentation, you have sexual reproduction in Basidiomycetes. Asexual nahi hai, but sexual is still there. So what happens with sexual reproduction is, so for sexual reproduction, what you need to remember for these Basidiomycetes are, they have a little similar to Neurospora, except there's two hyphae. They want to get lucky today. So there's two separate hyphae. What they do is they both create this type of, they both fuse at one point, plasmogamy takes place, and they create this type of dikaryon, a dikaryon body. It's a separate hyphae, it branches out, and it's dikaryon in nature. So it's N plus N in nature. So if I zoom in on one fragment, on one part, which looks like this, so there's one nuclei from A, one nuclei from B, one nuclei from A, and one nuclei from B. This is B, this is A. Okay, this is A, this is your B. So this is dikaryon. What happens here is this is called a hypha. Okay, the same thing, hyphae. And dikaryon stage, this also stays for a very long period of time. 100 years tak nahi jata, but it's a long period of time. Dikaryon stage for a long period of time. So it stays this way for a very long period of time. After this, what you get is your dikaryon stage takes place, then karyogamy will take place. You have your simple mushroom here. This is how a mushroom looks like. It has these gill kind of things inside. This is agaricus, the normal garden mushroom. So here, this whole thing is called as a basidium. Okay, basidium. What does a basidium do? Nothing. It's just a mushroom. So inside these gills, okay, these gills are the common subject here now. What happens here? These hyphae are inside these gills. You have your fragment here. So finally, karyogamy took place. Karyogamy takes place. After karyogamy took place, what you get is a diploid nucleus in the basidium. Okay. So after this, you get a diploid nucleus. Again, it's the whole story over again. Diploid conducts meiosis. So you get 4 haploids You get 4 haploid nuclei. So ये gill था. Inside this gill, karyogamy took place. After karyogamy, you got a diploid nucleus. It instantly divides by mitosis and gives you a haploid nuclei. After you get these haploid nuclei, what happens is not much. So it's still transversely, it's transverse here, but I'm drawing it this way because there's no space. So these haploid nuclei, they create something called a fruiting body, and spores form here. So just remember that they want to get out of the, out of the cell which it was inside, out of the cytoplasm. So it creates something called a basidiophore. A basidiophore or a basidiocarp. Same thing. It's a basidiocarp and it's known as your fruiting body. So it's known as your fruiting body. This is your basidiocarp. And the thing inside it is your basidiospore. So this thing inside, this is called your basidiospore. It's the same story all over again. This basidiocarp will burst. Your the spore will be let out. After this basidiospore is let out, what happens? This spore is let out. It germinates and grows into this. Okay, it will germinate and grow into this. This whole basidium is diploid in nature. Okay, haploid in nature. Basidium is haploid. Okay, diploid bodies are called sporophytes and haploid bodies are called gametophytes. So this is your gametophyte. N plus N कैसे आया? Dikaryon उसी तरफ से आया. This hyphae is N, this hyphae is N. They create something called dikaryon. Inside the dikaryon, karyogamy took place. Karyogamy took place, diploid nuclei came, diploid nuclei did meiosis. You get your 4 bodies. They create basidiocarp, basidiospore is formed, and the basidiospore germinates. So this is sexual reproduction in Basidiomycetes. We're almost approaching the end. So that was sexual reproduction in Basidiomycetes. Now you have your keywords in Basidiomycetes sexual life cycle. Basidiocarp, what we just did, it's the spore that's formed. It's the fruiting body which holds the spore. Dikaryon phase again, it's a pause phase between haploid turning to diploid. बीच में N plus N जो रहता है, it stays N plus N. Basidium is your mushroom, the whole body, it's called a basidium. And the name, what we call it is a mushroom, but the whole thing is called a basidium. This is your common mushroom, it's agaricus. Simple mushroom. Astilago and Puccinia. These are 2 types of Basidiomycetes and they create diseases called rust and smut in plants, okay, not in humans. So they cause diseases called as rust and they cause diseases known as smut. You have vegetative somatic cells, fragmentation. It took place from vegetative cells. Somatic cells is something that has nothing to do with sexuality. So somatic is just your body cells. So now my skin cell, which is there, nothing sexual is located in the skin cells. Okay, in your hair cells, in your eye, there's nothing sexual. So these are all your somatic cells. When you are learning DNA and genetics, usme you learn what is gamete and what is somatic cell. So gamete is your sperm, gamete is your egg, and somatic is all your other cells. So Puccinia, Ustilago, and Agaricus. These are your examples. You have your last type of fungi, which is known as your Deuteromycetes. It's called your imperfect fungi. So like I said at the start of the video, Deuteromycetes are a type of fungi which are not exactly like Basidiomycetes, like Ascomycetes and like Phycomycetes. Oh, they're kind of a different thing. They're imperfect fungus. There's no sexual reproduction only. There's no asexual, no sexual reproduction. So it's a very weird type. It's a very uncanny type of fungus. So there's no sexual, because there's no sexual reproduction, there's no sexual stage known for them. What I like to call them is the dustbin. Scientifically, it's not called the dustbin. What I like to call them is the dustbin of fungi kingdom, because when the first fungus you see, you have to see, does it have conidiospore, does it have conidiophore, does it have ascospore, does it have ascocarp, ascus hai kya? So this way, if you don't see anything from here, if you're observing correctly, and if you don't see anything here, you classify it as a deuteromycete. Their sexual stage is unknown. Once you finally, after a lot of observation, एकदम microscopic ascus दिखा, एकदम microscopic basidiospore दिखा, then it's known as then it can be classified into something. So Deuteromycetes में से you can classify it back. So right now, what fungal research goes on, it's in Deuteromycetes, in mycology. And their mycelium is also branched and septate. So what we learned from all of these classes is that the only one which has aseptate and cenocytic, unbranched, cenocytic, aseptate, whatever you want to call it, Phycomycetes में ही present है. Everything else is branched and septate. Basidiomycetes, Ascomycetes, and Deuteromycetes. Examples of Deuteromycetes: CAT, cat. So you have Collectotrichum, you have Alternaria, and you have Trichoderma. Trichoderma is a very dangerous plant fungus. So for us, what we get, जो एकदम life threatening होते हैं, we get moneron, we get bacterial infections, cholera, MRSA, all of these types of things are bacteriums. Viruses we get, coronavirus is going on right now. That's also a virus, COVID-19. So we get infections like these, and fungi have been known. All of these, which I said, cholera, these are a lot of fatal, MRSA. Then you have you have rabies. So all of this stuff, viruses, then you have your bacteriums. These all harm us, but fungi itna harm nahi karte comparatively. Fungal infection skin का सबसे ज्यादा दिखता है in humans, but for plants, rust, smut, trichoderma, these are fatal for plants. So these are the examples. This is how a deuteromycete looks like. This is a petri dish, microscopic होते हैं वो, and this is how it looks like from inside. So can you see here? Let me zoom in. This is interesting. So look at this one, okay? Look at this one. Look at this one. जिसका थोड़ा clear है resolution. Look at this one. I'll try to draw something like it. Can you see these small dots here? There it's haploid nuclei. And beach mein, if you look at other ones, you can see there's a little branching here. So what is this? Branched and septate mycelium. Done. So this is all there is. So we have approached the end of our video today. This is pretty long. The video was very long, but it's covered the whole fungal kingdom. There's nothing else fungi mein se which you can know more about. If you want to get into mycology, then you can learn more. But basically, NCERT ke liye, this is enough. If you're deciding to write NEET, or if you're just a casual viewer, if you're interested in biology, then you can go through this video once. This is for fungi kingdom. The next video which I will be creating is for viruses. I already created 2 videos on Monera and Protista, but there's some glitch with YouTube and they're not uploading yet. I will upload them, but this video it's uploaded properly now. It's premiered for 10 minutes. If you were here for the premiere, you would have seen it. Thank you so much. My socials are in the links. I have created a new Twitter account where I tweet continuously about science, about other stuff. And I have my Instagram. Thank you so much.